Okay. Let me show the screen and say that welcome to World for Women in Science. We are in the technical session for health session, and I would like to give the word to our moderators to start the session.
Yeah.
Hello, everyone. Hello, everyone. I am Professor Shaima Imani. I will be with you today with Dr. Kim Vaibi as a moderator for the health session. I hope you enjoyed our session today. So shall we start now, or we should wait for 2 or 3 minutes until other participants to attend?
We should start now. We are in the webinar already. Yes.
Okay. So we will start today with our first speaker, Dr. Kim Vaibi. Dr. Kim Vaibi is an emeritus professor. She is the president of Indian Association of Pathologists and Microbiologists. She is also vice president of Indian Association of Pathologists and Microbiologists. She is a chairman of Northwest chapter of IAPM. Today we will hear to her talk. I wish all of you to enjoy the session with Dr. Kim. The talk will be entitled Rare Complication Observed in Young Ulcerative Colitis Patients. So the mic is yours, Dr. Kim. You can start.
Okay, thank you for the kind introduction. Yeah, I hope you can hear me. Yeah.
Yes, we hear you.
Yeah. And so basically, I'm a pathologist, histopathologist for that matter. And we, as our institute, do caters to huge number of patients. So we do get to see many things. And because we have now mixed audience, the audience is mixed, it's not totally pathologist. So what I do is I think that I have to talk about ulcerative colitis as an introduction to many of you. And after that, I think just few slides, we come down to these two rare cases. I think that would be the right kind of thing which I decided to do. So this is my first slide and the title and myself. And so I have just broadly outlined my talk into 5 components. May not be very strictly, there could be a lot of overlapping. One is IBD. IBD stands for the inflammatory bowel disease and idiopathic ulcerative colitis. And this idiopathic ulcerative colitis, in short, we keep on saying IUC or ulcerative colitis. The complications of ulcerative colitis, we would be concentrating mainly in ulcerative colitis. The malignancy in ulcerative colitis, the malignancy, is it preventable in ulcerative colitis? And then after that, followed by just a brief presentation of those 2 cases, rare cases. Now coming to the inflammatory bowel disease. This is a chronic inflammatory condition of gastrointestinal tract, which is driven by inappropriate immune response to gut microbiome. Gut microbiome is coming up in a big way. Many diseases are being explained by the microbiome now, and there is a lot of genetic sequencing and so many things are going on about the gut microbiome, and supposed to be around 1.5 kilograms in our body. We all carry around 1.5 kilograms of microbiome within our gut. And this takes place only in a genetically susceptible individual. And broadly, this IBD can be grouped into, clinically, usually it is into 2, but morphologically to a pathologist, we can group this into 3. One is the ulcerative colitis, which is most common condition amongst all these 3. Second is the Crohn's disease. And the third is the indeterminate colitis. The third one, we make diagnosis only by histology. Clinically, there is no way for a clinician to say whether it is indeterminate or not. And usually, and so they would be treating these patients depending on the dominant clinical presentation as ulcerative colitis or as Crohn's disease. It's supposed to affect IBD is supposed to be seen in less than 1% of general population, but in our country, that is in India, some of the studies are quoting about 6%. So it's supposed to be increasing worldwide, the incidence. So it just depends on the kind of the study which we going to refer to. But this is a WHO data, and that is slightly older. So they have given us 0.4. So I have quoted that incidence. So individuals with inflammatory bowel disease, they have a tendency for development of neoplastic conditions, especially of the colorectal neoplasia, because they tend to develop dysplasia, and that usually tends to be followed by frank malignancy. And these are believed to be consequence of long-standing, ongoing chronic inflammatory conditions. So there is supposed to be an epigenetic change, which is associated, which is very important. It's not really genetic, but it's epigenetic change for the development of the cancer. And also they may have other rarer conditions of lymphoma and anal cancer. And also IBD patients usually tends to have increased risk of extraintestinal malignancy. So this is just to outline the difference, pictorial diagram to show the difference between ulcerative colitis and Crohn's disease. Ulcerative colitis, the mucosa is a continuous involvement. In Crohn's disease, these usually we call it classically as skip lesions. They may involve from one part to another part to anywhere. It's never in continuity. And these are very deep-seated inflammatory conditions. And so they tend to be associated with strictures. Now, coming back to the idiopathic ulcerative colitis, usually seen in individuals who are less than 30 years of age, though older people are not really exempted from getting the disease. Usually, as already stated, is associated or is because of the malfunction immune system to microbiome. And could be hereditary, but this is usually not very well documented, but usually tends to be seen in individuals with a background of family history of IBDs. It could be either ulcerative colitis or it could be Crohn's. And also individual who takes rampant NSAIDs, analgesics, medications, they are supposed to have more frequently higher risk of development of ulcerative colitis, and also individual who takes high fat diet, and people who tends to be very stressed out. And those are the people who are very prone to develop ulcerative colitis. And these particular inflammatory conditions can be controlled by changing in the lifestyle and also by medications. Control of flare-ups, that is the relapsing conditions. If it can be controlled properly, that reduces the risk of development of colorectal cancer. So controlling the disease flare is just not only to reduce the uncomfortableness it's associated with the disease, but also in the long run, it is to control or reduce the risk of development of colorectal cancer. So usually they come with bloody diarrhea, that is the main presenting symptoms. Others have uncomfortable abdominal pain, tenesmus, and also maybe sometimes if the patient is coming with very active disease with toxic megacolon, they may have fever as well. So this is the pathophysiology briefly, and I just want to draw your attention because of the inflammatory response, because of the altered immune system to the bacterial antigens which are present in the gut, that is could be either of dietary or bacterial in origins. Usually the release of the pro-inflammatory conditions, including TNF-alpha, interleukins, and these are the ones which are responsible for production of epithelial cell injury in the gut or in the colon, and leading on to the constant inflammation because bacteria and dietary antigens are constantly present within the gut lumen. That's why changing lifestyle becomes very important factor to control the disease condition. And so this is just a 3 patterns which one can see in ulcerative colitis. Commonness one is the one which are restricted to the rectum and the sigmoid colon, left-sided, right-sided, or pancolitis. Pancolitis are slightly rare. And so this is the histological features. Usually these are continuous mucosal involvement. Rectum and sigmoid colon are the most commonly involved. The ulcers have long linear ulcers, broad base, and pseudopolyps are very common features, and fibrosis, because it is usually mucosa and slightly extending to the submucosa. So fibrosis is not really seen in uncomplicated ulcerative colitis. And so this is just a gross photograph to show up the ulcerative colitis. These white spots, these are the normal mucosa, the remnant normal mucosa. So in between everything else is the ulcers. So diffuse ulceration in ulcerative colitis. This is a classic histological appearance. Cryptitis, cryptaphyses, where the neutrophil accumulation within the colonic crypts, lot of neutrophils within the lamina propria, mucodepletion. This is an active inflammation, very classic in a biopsy. And so this is what we call as pseudopolyps. So these are the pseudopolyps, and see the ulcers. This is a chronic long-standing case. So it's not really very active, not like the previous photograph. And that's why the color is becomes slightly more whitish. And so these pseudopolyps, which maybe sometimes like here, this becomes very kind of larger, nodular, indurated. So when one start having pseudopolyps like this, then one has to start suspecting it to be malignant transformation. So multiple histological examination, taking multiple section blocks is very essential to exclude any kind of associated malignancy, because the malignant need not be really protruding mass lesion. It could be just indurated flat lesion, which can only be identified by histological examination. So histological examination taken from multiple sites, even by colonoscopy, is a must and is very important. And so this is histological appearance. This is the chronicity changes, where there is crypt loss, crypt branching, crypt hyperplasia, and this is the pseudopolyp, where the mucosa is thrown into polypoidal structure. And so features of ulcerative colitis that associated with increased colorectal cancer risk. That means the colitis develop in young individual and difficult to control. And then extensive or pancolitis, and the patient has coexisting other morbidity like diabetes mellitus, and this appendectomy. This is a controversial. Some people say it is a protection. Some people say it's indicative of possibility of development of cancer. And so this is a kind of little bit of controversial, and also associated liver disease like primary sclerosing disease and presence of family history. These are the risk factor for an ulcerative colitis patient to develop cancer. So cancer incidence, according to the years, is 1%, 2%, 5% risk after 10, 20, and more than 20 years from the initial diagnosis of ulcerative colitis. Despite decreasing rate of colorectal cancer in IBD patients, but incidence rate is gradually increasing in young patients. So 10-year survival rates remain high, around 80%, which is in ulcerative colitis cancer. The outcome is not bad. It is similar to any kind of sporadic colorectal cancers. So intestinal lymphomas in the background of ulcerative colitis is very rare. Cancers are not carcinomas, are not adenocarcinomas, are not that very rare, but lymphomas are very rare. But compared to the other intestinal lymphoma, it is 2 to 3-fold higher compared to the person without predisposing ulcerative colitis. And so this is another pseudopolyps with larger polyps, and then there's a lot of flat indurated areas. So this is another receptor specimen. So when one start having this kind of confluent collision nodular lesions, one has to be very careful to exclude possibility of any kind of carcinoma, cancer, adenocarcinoma, or any other malignancy. So here one could see that these are the dysplastic epithelium, and this is the mucinous carcinoma. This is an adenocarcinoma arising in the background of ulcerative colitis. So why regular screening is essential to exclude colorectal cancer development in the background of ulcerative colitis, because earlier the states of any cancer is detected, the curative rate is much higher. And so if you're detecting a cancer at a stage 4, the curative rate is going to be very, very low. That is the reason that why all these ulcerative colitis patients should undergo screening depending on the type of the disease. We would come to that. And so this is an important factor for prevention of or to detect early colorectal cancer in the background of ulcerative colitis. And so ulcerative colitis, cancer risk. So depending on the site-wise, colorectum is the commonest. Then also they have higher incidence of liver and biliary tract, leukemias, other hematological malignancy, skin cancers, and they also have, these are usually related to the constant ongoing inflammatory condition. That means that disease activity, and which may go into toxic megacolon, and also person who has a coexisting primary sclerosing cholangitis with ulcerative colitis they are at higher risk for the development of cancer. And thiopurines, this is one medicine which has been very frequently being used, especially in Crohn's. This has been almost 100% association with development of cancer in IBDs. So nowadays, this particular drug is become almost obsolete, though it is a good drug to treat IBD. And so risk factors in ulcerative colitis, colorectal cancer development, there are three, one can divide into three factors. One is the disease-related factors, one is patient-related factors, another one is the histological-related factors. And so disease-related factors are the extent of the colitis. This is an independent risk factor. More extensive the disease process, higher the chances, the risk of development of carcinoma. So the cumulative index has already been, cumulative incidence, as I already stated in the first few slides, by 10 years, it is around 1% for the development of colorectal cancer. By 20 years, it's around 3%. And by 30 years, it is recorded to be around 7%. So these are because of the long-standing inflammatory condition leading on to the epigenetic changes. So this is very important. So a person may not really have any familiar or hereditary cancer, but because of the epigenetic changes, this cancer incidence of or higher, the patient can develop cancer. So when a person has concomitant sclerosis, primary sclerosing cholangitis, the chances of the risk of having a cancer is 2-4 higher. And also presence of strictures, anatomical abnormality in the colon. Also, they tend to have higher chances of higher risk of development of cancer. The patient-related factors are males are supposed to be having higher risk for the development of colorectal cancer. It is believed to be because of the protective effect of estrogen in women, and young onset of ulcerative colitis has been found to be an independent risk factor. Pathology related, as I already said, is the active disease which is ongoing and not really responding to the treatment. The patient tends to develop dysplasia. So routine screening is become very, very important to detect the dysplasia. And so especially if the dysplasia is high grade, on histology, the many centers usually subject the patient to pancolectomy, or depending on the extent of the involvement, they do resection of the colon. They do not leave the dysplastic or the high-grade dysplastic colon in vivo. They usually remove it in order to have a better prognosis. And so again, there is related to the severity of the inflammation, there is something called cumulative inflammatory burden. Nowadays, we calculate this in a given slide because these formula describe persistent inflammation in longitudinal cohort study refers to the histological inflammatory activity multiplied by the surveillance interval in years. So this is supposed to be a very good indicator for the development of colorectal cancer. So surveillance strategies for colorectal cancer in ulcerative colitis is first screening colonoscopy should be done at least 8 to 10 years after the onset of the symptom. High-risk patients should undergo annual screening by full-length colonoscopy. And the other risk factors are like a person with a family with a history of colorectal cancer in another family member before the age of 50 years, and a patient with primary sclerosing cholangitis, the presence of dysplasia, nowadays even low-grade dysplasia are taken as something to be definitive treatment, and presence of extensive active colitis with toxic megacolon. So intermediate risk factors, this is a controversial area, but they need to be a person who is supposed to have an intermediate risk factor. They supposed to undergo colonoscopy every 2 to 3 years. Low risk patients, possibly this is maybe once in every 5 years. So chemo prevention of colorectal cancer in ulcerative colitis, is it possible? Yes. Use of medical therapy, if the ulcerative colitis is controlled very well with medical therapy, then there is less chance or less risk of development of colorectal cancer. And how can I reduce my cancer risk? Suppose I have ulcerative colitis. Most important is healthy lifestyle, including healthy diet. And so there are certain factors which needs to be avoided and certain things which one needs to take more, like fish and all are supposed to be good proteins, but red meat, processed meats, these are, and soft drinks, fat, sugar, these are supposed to be avoided for person with ulcerative colitis. And now coming to the two cases which are in the index cases. The first case is person which is very interesting. Is a history of a person has hereditary spherocytosis who was diagnosed at 3 years of age, and he's been getting blood transfusion off and on, and he has been taking normal diet with normal growth. But for the last few years, he started complaining bloody stool off and on when he was 14 years of age, and he was diagnosed as ulcerative colitis, and he has been put on oral medication, but the response is supposed to be poor. Then recently he's come back with anemia, jaundice, and all, with the subacute intestinal obstruction. And when he is 22 years old, but his follow-up data are very poor, is not very good. This is what happens in our country. The follow-ups are very, very poor. Only few people, one could say that in India, we are one of the developing countries, so we have a lot of things, problems in treatment profile in conditions like ulcerative colitis, where regular follow-ups, screening, colonoscopy, everything are very, very important, but these are not really, cannot be really carried out regularly because the patient do not come back until and unless they have very severe problem. Otherwise, they do not really turn up. Very few people, very few patients would turn up. By now, he also has hepatosplenomegaly, cholelithiasis. So he has come back with many problems. And then he, as expected, his hemoglobin is low. TLC was high, total count with leukocytosis, viral markers were negative, G6PD screening because he is basically abnormal hematological profile. So these were all done and they were found to be within normal study. CECT abdomen showed inflammatory thickening of ascending colon and the cecum, and also his was circumferential mural thickening in the hepatic pharynxectomy. He had multiple site thickening of the wall of the colon and also massive splenomegaly. This could be because of the hematological associated hematological problem which he has, the splenomegaly, may not be really anything to do with the cancer or something to do with the ulcerative colitis. So colonoscopy was carried out. It was stricter at 60 centimeters, and he had a florid disease, multiple pseudopolyps, nodular areas, edema, loss of vascular pattern. All the abnormalities were present in this particular young patient. This is one of the colonoscopy picture. And so he underwent open surgery and resection, and there was 4 into 5 centimeters circumference of growth was identified in the mid-transverse colon, and along with multiple enlarged mesenteric lymph nodes and numerous pseudopolyps and diffusely inflamed mucosa and pectoralis IC valve. And this is just a part of the resected specimen with the lesions somewhere here. These are the lesions here. One could see that is a grossly abnormal. This is unfixed area actually, so it's become slightly reddish. So microscopic showed a below glandular pattern of adenocarcinoma infiltrating up to the subserosa. So, and he also had a CMV infection that could be just coincidental because he was already immune compromised and he was malnutrited, and so that could be CMV flaring. Because all of us carry CMV in our body, especially in the colorectal mucosa. It remains redundant. We all are exposed to it only when our immune status becomes compromised and it flares up. So this CMV could be just localized infection, need not be really systemic infection. And so that was the diagnosis in this. This is the histology of the same tumor. I think there was some more photograph. I think it has not come up. The low power photograph, but it's okay. This is the high power to show the infiltration into the smooth muscle. These are the tumor cells arranging glandular pattern. And so this is the tumor on the mucosa and also gone to the surrounding fat. So pericolic fat has been extensively involved. So it's very advanced stage tumor. And adenocarcinoma was made, and he was on follow-up treated, but I think within 2 years' time, he succumbed to his illness because he had very, very severe malnutrited child with very advanced stage tumor. So there was nothing could be really done. So the second case is another 24-year-old male. He had similar kind of loose stools, abdominal pain, and off and on for 2 years, he was not really gone to any proper gastroenterologist. He was by local medical practitioner. He was given some antibiotics, but there was no proper response. And later on, he was taken to an institute and he was diagnosed as ulcerative colitis and was given treatment, but with poor response. And so diagnosed as refractory ulcerative colitis. So lab investigations, more or less, there was nothing really very significant except for anemia and leukocytosis. Colonoscopy, again, there was severe involvement of the rectum, and a full-length colonoscopy was carried out. Again, there were a lot of ulcers, erythema, pseudopolyps, everything, and the patient underwent. There was no definite tumor was identified, though there were some flat areas were identified on colonoscopy. But because of the severity and presence of toxic megacolon, the patient was subjected to a subtotal colectomy. And so entire colon was involved and grossly inflamed mucosas, and there were some nodular areas and some moral thickenings. So multiple random sections had to be taken all the time, and every section has to be examined because there could be a lot of variation in the histological findings, and there could be just one focus where you have the tumor, and so all the sections need not have, so one cannot really leave one section alone. You have to examine every section, 10, 20 section, whatever section has been taken, that has to be examined. And so this is the histology where the mucosa is grossly ulcerated, but along with it, there's a lot of infiltration which is gone deep into the muscle and serosa. This is the high power. These are, I don't know whether all of you all could make out, these are all plasma cells, sheets and sheets of plasma cells on high power. And so this is a few of the immunostains which were carried out. CD38, 138 are plasma cell markers, and CD20 and T cells were just scattered cells were positive. They were lambda was positive, so kappa were restricted. So it is a kappa restricted plasma cell, plasmablastic lymphoma. These were all high grade, and regional lymph nodes were also involved. And this lymphoma was multicentric. It was not just localized in one area. So it is an extensive disease with poor prognosis. And so this is a diagnosis, the final diagnosis in this case. I think that was my last slide. Thank you very much for your kind attention. And I think this is my last slide. Any query, anything? I think I would be ready to answer.
Thank you, Dr. Kim, for your nice presentation. It's very interesting. So now for the participants, is there any question to Dr. Kim?
For now, there are questions in the question and answer section, but if there are, we can take them at the end of the session.
Okay. For me, I have one question, if please, Dr. Kim.
Yeah, please.
I would like to ask you about the microbiome for the colorectal cancer patients. Did you study exactly the profile of the microbiota for the colorectal cancer patients?
Not in this, we do not really do, because these should be done taken up by the microbiologists. I also know that there extensive works are going on. So as a pathology, histopathology, we do not really do. In our center, you know, pathology, 4 departments, a huge big center we are. This is a PGIMER in Chandigarh, a huge big center. We are more, it is 12,000 bedded hospitals with many advanced centers. So even in pathology, we have 4 departments: histopathology, immunopathology, cytopathology, and lab hematology with all DM degrees. So we, you know, and then we have a huge big microbiology department. They have again virology, bacteriology, mycology, you know, they do extensive work. So we do not really encroach into that. We do not really have time. I know that you would be interested, but there are a lot of applications. So may go into this, and many people are working. Actually, microbiome is coming up in such a big way. You know, we do not really study much about bacteria as such as the histopathologist.
I see. So because I am a microbiologist, so I am interested to know
I know
which
that you
type
would
of
be
bacteria
interested.
will be associated
Yes,
with the colorectal
definitely.
cancer.
Yeah, I understand that. I expected also.
Yes. Another question is about the surveillance strategies. Did you suggest some surveillance strategies for preventing the colorectal cancer?
Pardon, you repeat again, please. It was breaking up your voice.
After the conclusion in your study, did you release some surveillance strategy?
Surveillance program. Yeah, surveillance program. Actually, you know, we don't do it in normal people. That surveillance program which I was talking about was for ulcerative colitis patients. And so whoever, we all the time ask them that you come back after 1 year, you come back after 2 years, depending on the disease activity. Because otherwise in India, it's not possible to do surveillance. You have to just see our hospitals. It's madness. So many, we can't do it. That is only the disease people. So getting into the general population, usually it is done by the community medicine people, and also from like we have huge government funding agencies like ICMRs, DBTs, DST. They take it up separately, you know, like institute like us, until and unless somebody like community medicine people do it. The rest of us is almost impossible because patient load is so heavy. We can't go to the community. So very, very different story we have to tell you in our country. We huge population in India. And Dr. Ritu Parna is there. She would be also aware. She is working in one of the, I think, you are in DPT or DST?
Okay, thank you. We have another question.
ICMR Institute.
ICMR. So they all do the funding. And these people are the funding agency.
We have the last question. is the surgery enough to solve the problem?
Pardon?
One of the participants asked about the surgery. Is the surgery enough to solve the problem of colorectal cancer?
See, surgery is just reducing the risk, or I mean, like the chances of metastasis, further metastasis. It depends on the states of the disease. Suppose you have a disease at stage 1, stage 2, it is curative.
Okay.
So the earlier the surgery is done, in stage 4, where there's 50 lymph nodes are involved, pericolic fats are involved, then surgery may reduce or extend the lifespan of the individual with the chemotherapy and post-op, maybe 2 years, 3 years, that's all, not more than that. So the early stage surgery is for early stage to treat as a treatment. Okay, surgery should be done. It is very good prognosis. Stage 1, stage 2 treatment, colorectal cancers are treatable, curative with surgery. Yeah, so it just depends.
Okay.
Any tumor, it depends on the stage.
Yes. Okay. Thank you very much, Dr. Kim,
Thank you
for your
very
nice
much.
presentation. And now we will move to the second speaker, Dr. Ritu Paranadi. I hope I
Ritu
pronounced
Paranadi.
the name.
Yeah,
the
I know it's
pronunciation is
difficult.
correct.
Even I was asking you about yours.
Yeah. So
Evening,
Dr. Ritu Purana is dedicated to the cause of infectious disease research and studies. And today she will present about the identification of prognostic markers and probiotic candidates by screening of the gut microbiome in an endemic population of the area. So the mic is yours, Dr. Rita.
Thank you. I'll present to you. So I'm going to speak about the identification of prognostic markers and probiotic candidates. Just a minute. Sorry for the interruption. Okay, so I'm going to speak to you about the identification of prognostic markers and probiotic candidates by screening of the gut microbiome in an endemic population of diarrhea. Actually, we did this study in compliance with SDG 2 and 3. And as you know, the diarrhea is one of the major contributors of mortality, both in adult and children. And in under age, totally taking the entire population, globally, it's the 9th major cause of mortality. And in under 5 children, it is the 2nd largest cause of childhood mortality. And these are the figures that you see from the World Health Organization. And it mainly affects the poor, the poverty-stricken population in the low-income countries and the low-middle-income countries, mainly. And these are the parts of the world that fall mainly in Africa, Southeast Asia, and Latin America. And these are also endemic regions for diarrhea. So, it contributes to a lot of socioeconomic loss. It leads to malnutrition. And the relationship with malnutrition is also very interesting because malnutrition, it impairs people's immunity. It makes them more prone to diarrheal infections. And again, diarrhea will deplete all the nutrients from the individual's body, and it will make the person more malnourished. But at the same time, diarrhea is prevented. It is by if we administer proper hand washing and proper hygiene and safe and fecal matter free drinking water and good sanitary practices, diarrhea is prevented. If anybody is affected with diarrhea, it is also reversible by administration of ORS, and even 20 mg zinc and antibiotics are kind of the treatment options for diarrhea. But according to the GAP-D program of WHO that stipulates protect, prevent, and treat, the best option would be to prevent diarrhea. And for preventing diarrhea, an early warning system is very, very important. It is in place already. Surveillance studies are very routinely done. And the institute I'm working in is completely dedicated to diarrhea research. Here we do have a unit completely dedicated to diarrheal surveillance. And usually we use, traditional measures like phenotypic measures or phenotypic methods or genotyping, the traditional genotyping techniques for surveillance, for detection and diagnosis and for surveillance. But lately, WHO has also preferred NGS, the next generation sequencing methodologies for epidemiological surveillance. So my goal was, if NGS can be extended, the benefit of NGS can be extended beyond just serving as a surveillance method, as an epidemiological surveillance method, to also aid in research goals related to prognosis, enhancing prognosis of diarrhea, or looking for even probiotic candidates or some kind of therapeutic candidates for assuaging the burden of diarrhea. So we focused on comparison of the diarrheal and the non-diarrheal microbiome in order to identify these, if we could find any potential candidates, like bacterial candidates, which could serve as prognostic markers or even therapeutic markers, which could be developed as potential probiotics, and they could be administered to this malnourished population as a supplement, dietary supplement. So that also complies with the SDG 3, SDG 2 and SDG 3. So the major goal, I will first, Dr. Vaipe has already mentioned about the microbiome, but as we all know that every ecosystem on Earth is infested or rather is inhabited by different microorganisms, and they are involved in different functions, beneficial functions, and that forms a microbiome. Similarly, our gut also has trillions of bacteria inhabiting and forming the microbiome. The gut, it starts with the mouth and it ends with the anus, and almost every part of the gut has been found to harbor a microbiome. And this microbiome actually, it starts developing prenatally, like even before birth, from the mother to the offspring it comes, and it is kind of established by the age of 2, and then it keeps changing with time, and it depends a lot on the factors that affect the microbiome and its development are genetic factors, diet, lifestyle, and the geographical location, and many other factors. And the microbiome is very crucial for maintenance of, health, a healthy state of the body. They're involved in very vital functions like physiological functions, metabolic functions, according immunity, homeostasis. And it has been found that when an individual is affected by any disease, be it infectious disease or chronic diseases, this microbiome composition is disturbed, and this is what we call dysbiosis. So initially, in a pilot study with just diarrheal samples, I found that it was the healthy microbiome, the healthy bacteria in the microbiome, they had got like kind of suppressed, and it had given way to enrichment of the bad bacteria, like the opportunistic pathogens, or the mainly the opportunistic pathogens, and those which are really harmful, and that is what we label as bad bacteria. So this time I conducted a study where I also incorporated healthy samples, healthy fecal samples, and compared it with the diarrheal fecal samples. So the study was mainly designed to examine the dysbiosis occurring in diarrhea, to identify taxa that could be used as I already said, prognostic and therapeutic markers, with which the prognostic markers, or the therapeutic markers, could revert dysfunctional gut by restoring normobiosis and promoting eubiosis. So the major goals were differential identification and also discriminating between the diarrheal and the non-diarrheal microbiome in terms of diversity, abundance, and also identifying taxa which could become very sharply associated with diarrheal dysbiosis, and then kind of working out a statistically honestic correlation between the communities of commensals, and because microbiome is composed of communities of commensals and opportunistic pathogens. Opportunistic pathogens, usually they will not harm the individual, but in case the commensals go down in abundance and in number, it is then that it has been found that opportunistic pathogens, they tend to show a pathogenic effect. Methods that we applied. So this was a cross-sectional gut microbiome study analysis, which comprised 23 non-diarrheal and 5 diarrheal fecal samples. They were collected from urban parts of Kolkata, and we sought informed consent from the subjects before collecting the samples. Then we use 16S rRNA amplicon sequencing. That's a method of next generation metagenomic sequencing, where we extract the stool DNA, the entire DNA from the stool, enrich them for the bacterial DNA, and then we carry out sequencing for the 16S region, the conserved 16S rRNA or DNA encoding the 16S ribosomal subunit is very conserved. So it is also, it comprises conserved regions and also variable regions. So this variable region is sequenced in order to show the taxonomic differences or diversity of the microbiome. And it was sequenced on the Illumina MySeq platform. And then we use the Green Gene database for taxonomic identification and annotation. And then we worked out the relative abundance of different phylum and families. And we are mainly interested in the level of phylum and families, because beyond this, it's a bit difficult to identify the OTUs till the genera or the species level using this, using Illumina, this method of next generation, because the other alternative method, the whole genome shotgun sequencing, is better option for identifying till the genera or the species level. And then we calculated B by F ratio, because this is like Bacteroidetes, the phylum Bacteroidetes, by a ratio between the Bacteroidetes phylum and the formicutes, because in case of disease, we find that formicutes are enriched, and in case of a healthy state of the body, we find that formicutes are suppressed by the Bacteroidetes. So, and then we represented the difference in mean relative abundance using different histogram and stat bar diagram, and we conducted statistical analysis on these statistical analysis. So at the family level, we found that these 4 families, Actinobacteria, Proteobacteria, Formicutes, Bacteroidetes, they were all consistently found in all the samples, but their abundance differed. The Formicutes was the most dominant phylum in the both groups, but we found that Bacteroidetes were dominant in 43.5% non-diarrheal, but they were not at all dominant in diarrheal samples. So, and the average B by F ratio was consistently found to be less than 1, showing that Formicutes was more abundant compared to the Bacteroidetes. And we found that Proteobacteria was overrepresented in the diarrheal samples, and only 3% was found in the non-diarrheal samples, and the difference was found to be statistically significant. You can see in the in the graph. And same thing we found. We also found that crucial, very significant differences at the level, family level. So we found that Lacnospiraceae, Erysipelotriaceae, Privatulosae, Streptococcaceae, Enterobacteriaceae, like especially these Privatulosae is a family of commensals, whereas Enterobacteriaceae, Streptococcaceae are the more pathogenic and opportunistic pathogenic families. So these were found, but significant differences were there. Like we found that Privatulosae was the most abundant family in majority of the non-diarrheal samples, and they were significantly suppressed in diarrheal samples. Then we found that Streptococcaceae was the most abundant family in 60% diarrheal samples, and where Streptococcaceae was like suppressed in the non-diarrheal samples, we found that Streptococcaceae was most abundant family in the majority of diarrheal samples, and then Bacteroideaceae and Nocardiaceae were the most abundant when Streptococcaceae was suppressed. And in the non-diarrheal samples, Streptococcus was completely suppressed by bifidobacteriaceae. And then we found a negative correlation between commensals and, as I said, families of pathogens like Streptococcus and Enterobacteriaceae. And at the species level, we found that Proteus mirabilis was associated with all the non-diarrheal stool samples. And it was very interesting because they were completely absent from all the diarrheal samples. So we got a glimpse into like what could serve as a prognostic marker, or it could also even be developed as a probiotic candidate. And then Prevotella copri was present in both the groups, but it was more enriched in the non-diarrheal. And the difference was significant. So coming to the discussion, I would like to point out that we found significant negative correlation showing that the substantial antagonism exists among the communities of the commensals and the opportunistic pathogens, which could be actually exploited for reaching our goal, like Lachnospiraceae and Enterobacteriaceae. And we aim to take this up for our future study for going in the direction of enhancement of prognosis. This could also serve as a prognostic marker for screening vulnerability, for developing an early warning system in the endemic regions to tell us whether, if we screen the population and if we find a similar kind of composition and abundance difference, it could tell us whether the population could be more prone to diarrhea or not. Then because we found that bacteria like Bifidobacteriaceae family, it suppressed pathobiome, so these could also be developed in endemic population. The Bifidobacteriaceae could serve as a marker, or it could, Bifidobacteriaceae is quite a well-known probiotic candidate. It could be even used as a probiotic. So as I already said, that we are thinking about developing Prevotella copri mainly. We could identify this species. And the findings were reinforced by epidemiological data that the frequency of occurrence of these markers was significantly different in the two groups. So we aim to develop these as the prognostic and the therapeutic markers. So in conclusion, this is actually the first report on the comparative analysis of diarrheal and non-diarrheal gut microbiome, in which we identified prognostic markers, prospective prognostic and probiotic candidates for which could accord protection to the endemic population. This would benefit particularly the economically backward areas of the world where gut microbiome dysbiosis due to different parameters like malnutrition makes them vulnerable to the diarrheal pathogens. This could be administered as a dietary supplement. And so the key messages from this study could be that new age technology like next generation sequencing is a very potential tool for the detection, prediction, and prospective development of prognostic and therapeutic markers, and that the gut microbiome holds tremendous prospects for the development of novel probiotics and prognostic markers, and the research methodology is unique and novel. It has this way, this technique has not been exploited so far. So I ventured into doing it, and I found quite prospective results. And it is also prospective for understanding the gut microbiome from a critical vantage point, which led to the identification of prognostic and therapeutic candidates, which can help to revert dysbiosis and establish normobiosis. These candidates may be administered as dietary supplements and also could take us a step ahead towards the SDG 2 and 3. Especially by administering them in the poor endemic population, which is vulnerable to diarrhea. So I would like to thank the Department of Health Research and ICMR for supporting this work, and thank you.
Thank you, Dr. Ritu Parana, for the nice presentation. And now we have one question about what is the link between malnutrition and diarrhea?
Malnutrition and?
Diarrhea.
Diarrhea. Yeah, because see, if the population is malnourished, which means like they are more prone to diarrhea because they don't have that immunity. because because nutrition is required for development of immunity. And here we find that children, mainly the children under 5 and also children, they suffer from malnutrition and they have impaired immunity. So they are more prone to the attack by diarrheal pathogens, because to resist any kind of infection, you need, solid immunity. And again, once we are infected with any diarrheal pathogen, we lose all the nutrients from our body also. So that way, the person becomes weak. So it is like a cycle.
Okay, thank you so much.
Can I ask you some questions?
Yeah, sure. I would.
For maybe that I missed out. So how many samples have you studied, and what were the source of the samples? And then that was number one. And number two would be, before going to the human beings, do you want to try it out in animal model so that it has to be approved? You just can't go straight to the human beings and start giving them this. There are many agencies to prove it, to overlook that, and you have to get it approved. Yeah. So what is the steps which you'd like to take up for? There are two major questions. Okay.
The first answer to the first question is they are all diarrheal stool samples, and also stool samples collected from the healthy population in Calcutta. So we actually totally we studied 30 diarrheal samples, but in this comparative study we studied 23 non-diarrheal samples and 5 diarrheal samples for the comparison I used. So they were all.
Do you feel that, sorry, to disturb you, do you feel that studying only in 5 diarrheal sample, do you specify the kind of the diarrhea, duration of the diarrhea, age of the patient? There are many factors which you have to look into. You just can't take the sample and start studying it.
No, they are like all acute diarrheal patients. And they
5 is very less. 5 is very less. It has to be in hundreds.
Yeah, you're correct. But actually, as I said, I first conducted a preliminary study using 25 diarrheal stool samples, and there we found significant, the results. And then we compared those 25 with 5 of these, their profile, and we found that the comparison was true. So for this comparison of non-diarrheal and diarrheal, so we incorporated these 5 for the comparison.
What about, you were planning to go to the publics. Before that, what are the things which you plan to do? Going to the human beings, before going to the human beings.
This is a clinical survey. So first, in order to get the real-time data, we have to do in the human population, the diarrheal patients and the non-diarrheal. So now we have our findings, and now I'm testing these in the laboratory. First, I'm doing the studies, the commensal and the antagonism relationship studies in the laboratory between the commensals and the pathogens using their biofilms, and then I'm going to actually see the effect of these prospective candidates on cell lines and also using organoids first. And then only we can do if it's something. I'm hopeful that something good will come up, and then we will test it in the human population.
No trial in animal models?
Yeah, of course.
That's very important. Before going to human beings, you have to have the animal models in between.
Yeah.
You can't skip that.
Yeah. So in diarrheal animal models, that the rabbit ileal loop is there. So we will do that.
Right. There are a lot of ethical issues, right?
Yeah.
Yeah. There are a lot of human rights, animal rights, a lot of things which we have to go through.
Yeah, very difficult to also get ethical clearance for animal studies.
Yeah, anyway, well tried. But 5 samples, very less. You have to try it in many more.
Okay.
Okay, thank you for your presentation. Actually, we ran out of the time and we still have 3 presentations. So can we delay the discussion for the end of the session? Because now we have many questions and still there are 3 speakers. Thank you so much.
The next is yours?
Yeah,
the next is yours? The next is yours?
I will be the
next
I'll
introduce
speaker.
you. Yeah, the next is yours. I introduce you, right?
Yes.
Shall I introduce you? Okay.
Yes, please.
So now the next speaker is Professor Inani. She is from Egypt, Cairo. And her biodata speaks volumes about what kind of work she has been working. I'll just, and she has been awarded many international as well as national awards, scientific awards. And she is also holder of the World Academy of Sciences, that was Young Arab Scientist Prize 2018. That's a remarkable award. Yeah, I am just naming few of them. Otherwise, there are many things. And also she is a member of the Scientific Committee of World Forum for Women in Science. Okay, and also member of the Global Open Science Group. And then I think I would just like to mention as the last, and she had been chairperson and also co-chairperson for the COVID diagnostic group in Africa, and also for low and middle income countries. These are big achievements. There are many more, many things are there. I think we cannot keep on going on. And so with these few introduction, I think I will request you to start your speech. I mean,
Thank you for the
your
introduction.
lecture.
Thank you for your introduction.
Yeah.
Actually, I would like to ask for a share screen because I'm not allowed to share my screen.
No, that share screen down there, green one. You open your PowerPoint first. Please open
I
your
already
PowerPoint
opened it,
first.
but
Then
I
after
think
that share screen, green thing is
Ritu
there,
Puran,
down
did you
there.
stop?
Achha. You open your
It doesn't
PowerPoint,
work.
please. Achha. You can do it again. Open the PowerPoint, and after the share screen, that green thing,
I
open
already
your
opened it.
PowerPoint first.
I'm not sure. Rita Puran, did you stop the sharing?
That share screen, okay.
Stop again. You're a co-
Okay, so thank you for calling, huh? हाँ थैंक यू फॉर कॉलिंग। मैं कुछ वेबिनार में हूँ, कुछ पूछना था, हो गई, मैंने नलिनी को पूछ लिया था। ठीक है, थैंक यू, थैंक यू, आई विल कॉल
Did
यू
you stop, Ritu Puran, please?
Yes, she stopped the screen sharing, and I made you a co-host, so it should work.
Yes, please, because I'm not allowed to share my screen.
And when you press it now,
Can you do it? Or otherwise, we invite the next person, then you can do it later.
Okay.
It's done? No? I think the technical staff, we had that lady, you know, she will help you, I think.
Okay, now I think it will work.
Yes.
Can
Now
you
we
see my screen now?
Yeah. Yeah, slideshow. Now you go to the slideshow.
Is it okay for you now? You can see?
Yeah. Slideshow. Click the slideshow down there. Click the slideshow, this one. Niche, down. Click the slides.
Yeah, okay.
Click there. I think it's not coming on. It's coming. Yeah, full screen. Then you can do full screen.
Yeah, okay. I'm sorry for
No issue. Yeah, you can do the full screen.
Okay. So today I will talk about the first emergence of colistin gene MCR10 and fosfomycin resistant gene FOSA5 in Klebsiella pneumonia. This is the first emergence in Middle East. The aim of our study was to investigate the occurrence and resistance of different phenotypes from gram-negative bacteria in mastitic cows and unpasteurized milk. Also to identify the multi-locus sequence type, resistant genes, and the plasmid replicon in these types of bacteria. To determine the genetic characters of different genes like MCR10 and fosfomycin 5. To detect the epidemiological relation between our isolates and the public available genome for the Klebsiella pneumonia that harbor the same gene, MCR gene. For this objective, we isolated and identified gram-negative bacteria from clinical and subclinical bovine mastitis and from raw unpasteurized milk to investigate the prevalence of these bacteria, of gram-negative bacteria, and investigate the antimicrobial resistance phenotypes and also the genome sequencing for selected gram-negative bacteria. Our results indicated that we identified 100% of Klebsiella pneumonia, Pseudomonas hydrophila, Enterobacter cloaca, and all these are identified in bovine milk, in the unpasteurized milk. We identified also around 87% of Escherichia coli and around 70% of Proteus mirabilis. And this is where the most prevalent multi-drug resistant species we found in the milk. Not only multi-drug resistance, but also we found extensive drug resistance phenotypes of these bacteria, like Proteus mirabilis was around 30%, and Escherichia coli 3% were isolated from the milk. 10 isolates like Escherichia coli, Klebsiella pneumonia, and Proteus mirabilis displayed the highest multiple antibiotic resistance and exposed to the whole genome sequencing. So we selected the samples which will be sent for the whole genome sequencing based on the multi-drug resistance, the bacteria which contain the highest antibiotic resistance gene. Our results showed that we identified in the Escherichia coli 2 multi-locus sequence types, sequence type 2165 and sequence type 7624. Sequence type 2165 will show similar profile of the plasmid replicon INC-FIA and INC-FIP, INC-FII, INC-Q1, with the exception of only isolates containing INC-R. For the sequence type 7624, we showed a different plasmid profile, including INC-HI2 and INC-HI2A. Using the risk finder tools or risk finder websites, we revealed the presence of plasmid-mediated colistin MCR10 and fosfomycin fos-A5 resistant genes in Klebsiella pneumonia, which is isolated from bovine milk. We did the sequence analysis for these bacteria which containing the MCR10 and fosfomycin A5, and also we did the plasmid sequencing. The sequence analysis of the reconstructed MCR10 plasmid revealed that the MCR10, as you can see in the figure, is bracketed by XERC and the insertion sequence 26. So it's located between both of them. Also, our finding reveals the clustering of these isolates in a clade containing isolates from human, from environment, from animal, from different countries like China, Singapore, Thailand, United States. For this result, it's very important because, as you can see, we isolated the MCR10 gene bacteria from bovine milk, but different countries found the same gene in humans, others in environment, and others in animals, which indicate or emphasize the concept of One Health. So these genes could be transferred from human to animal, from animal to human, from human to environment, from environment to animal. So it emphasizes the concept of One Health and indicates the importance of using the antibiotics in both animal and human, besides the environment also. Again, the phylogenetic analysis for these isolates revealed the existence in clade containing MCR10 harboring genes from human environment with different sequence types and also isolated from different countries, which is indicating the spreading of these genes among different isolates and in different countries. In conclusion, we reported the first emergence of MCR10, which is responsible for the colistin resistance, and fosfomycin A5 gene, which is responsible for the fosfomycin resistance in bovine milk for the first time in the Middle East region. And this conclusion emphasizes the importance of the concept of One Health. It constitutes a public health threat. And here is the penetration of the last resort antibiotics because, for example, colistin was considered one of the last resort antibiotics which is used for treating human. And also the use of antibiotic in both human, animal, and antimicrobial surveillance plans are urgently required. Thank you very much. And if there's any questions, I will be happy to answer.
So wonderful work. Yeah, I think I take the advantage to compliment you. And have you published a paper, this one?
Yes,
is it published?
we already, yeah, we already published 2 papers from this work.
Okay. So you are going ahead and studying in many more samples? You're extending your work?
Actually, we isolated around 300 samples from milk and 300 samples from mastitic bovine mastitis.
Okay. So you're planning to expand with the like correlate with the woman, I mean, like the human beings in the surrounding areas, in the area, locality?
Yes, I think the most important step next to this work to isolate samples from hostels, from human inside the hostels, to find the correlation between spreading from animal to human, from environment to human.
Yeah, environments. Yeah, good. I say it's going to be. You may come up with a big breakthrough. Yeah, of the I think antibiotic resistance. That is a big issue. I think everywhere, all throughout the globe. I think that's a big problem for the clinicians to handle it. I think you must continue, and I think I really appreciate it. It's a fantastic work.
Thank you so much. So I
Any
think,
more
Rita,
questions?
you have a question. Rita Paran, please go.
Yeah. I think Ritu Purna has a question.
I was just inquisitive that the K1 plate that you found and those samples that you collected and they fell in the K1 plate, you had collected all from the same time span, or they were like temporarily
No, the same period. It's around 6 months to collect all the samples.
Okay. And have you tried to trace the origin of this marker, this plasmid, the origin of this?
The origin? Actually, what we concluded in this study, it's an acquired resistance gene, maybe because in Egypt, they use the colistin in the food of the animals. So maybe this is the source of the transmission of the colistin gene
Animals,
from
zoonotic.
Yes.
Okay. But which country or which part of the world have you tried to see? Like it's indigenous, it's endemic to Egypt, or it's come because we frequently report this in India also. Like, so have you tried to see from where this has come into Egypt, or?
Actually, for this study, we isolated the samples only from Egypt, but we detected the emergence of these two genes for the first time in the Middle East region. The correlation between these genes and other similar genes in different countries, we only exported the genome from the internet and compared with different countries. For India,
cool.
I'm not sure. We didn't find from India, but we found in United States, Singapore, Thailand, and many other
like
countries.
You should. Yeah. Mm-hmm. Thank you.
Welcome.
Any more questions? Anybody from the panels, from the speakers? I don't think there is any question in the chat box. Okay, I don't know. There's one thing. Sorry, Ritu Purna. I think, okay, you're typing the answer? Okay, fine, that's fine, I think. Otherwise, I think we can go ahead to the next speaker. Yeah, who is the next one? I think YM Kyumika Yapa, right?
Yes,
Okay.
Dr. Kim.
Yeah, you would be the next speaker? Okay.
Yes, Dr. Kim.
So, hello? Yeah, good evening, good evening. Are you from which country? From Sri Lanka, right? So good evening. So you are a very young person. How old are you? You compared to us, you are much younger. So she's working towards sustainable future through nanotechnology-based findings. Huh? And so you are a PhD scholar from Sri Lanka. Yeah. And so I think you are doing a lot of research works using nanomedicine, nanotechnology, everything is in nano. And so with this little bit of introduction, I think you please go ahead and start your talk.
Okay, thank you, Dr. Kim. Shall I share my screen?
Yeah, please do.
Okay. So I think I'm audible and my screen is visible to you.
Yeah, definitely we can hear you and we can see your screen.
Okay, good afternoon, all of you. I'm Pyomika Yappa. So I'm going to discuss about my research findings of our study on synergism of multimetallic nanohybrids as promising antimicrobial agents with an expansive coverage of microbes. So without further delay, let's move on to the introduction. As all of us know, bacterial infections have now become a serious threat to the public health. The main reason behind this tragedy is antimicrobial resistance. According to the recent reports, nearly 5 million deaths was caused because of this antimicrobial resistance in 2019. Also, when considered the number of deaths due to the bacterial infections in 2019, 1/9 were caused because of methicillin-resistant Staphylococcus aureus. So this data clearly shows us some demerits of the conventional antimicrobial agents like antibacterial resistance to conventional antibacterial agents because most of the microorganisms shows a number of adaptations to these conventional antimicrobial agents and also prolonged illness and hospitalization, high mortality and morbidity rates, and all of us know they are somewhat expensive, and also the narrow spectrum of the antibiotics, which means current antibiotics are narrow spectrum, which means one antibiotic is specific for one type of microorganisms. So it is urgent to invent and imagine antibiotic agents to overcome these obstacles. So as we know, metallic nanoparticles have a well-proven antibacterial, antimicrobial potential, and they show a minimum antimicrobial resistance. And owing to their small size, which means scale in nano dimensions, result a high surface to volume ratio. They are relatively cost-effective when comparing with the conventional antibiotics, and also they show unique defense mechanisms against the structure and physiology of microorganisms. And also they are relatively cost-effective, and their synthesis process is straightforward, which means they are somewhat cost-effective when talking about their synthesis process and comparing with the massive R&D process of this manufacturing of conventional antibiotics. And here we have chosen 3 metallic nanoparticles. They are silver, copper, and cobalt nanoparticles, and we have doped those nanoparticles into a silanol network in silica nanoparticles. So why these silica nanoparticles? Because silica nanoparticles is an excellent material as a cargo loading agent because owing to its high porosity and also the diameter of these pores are homogeneous and the surface area is also very high. The volume can also adjustable according to the desires of the researcher, and they are apparently non-toxic and they are biocompatible and biodegradable. These are advantages when we are talking about biomedical applications of these silica nanoparticles. And the main point here is the silanol network can be doped with various metals. This point is the main point of our research. So our main objective is to synthesize, characterize, and compare metal-doped silica nanohybrids as potential antimicrobial applications. So why these synthesized silica nanohybrids are significant? Because they can be combined to formulate multi-metal-doped silica nanohybrids, and also this approach will enhance the synergistic antibacterial potential. And also they can be used to target a broad spectrum of bacteria, including gram-positive and gram-negative bacteria. And also they can be incorporated easily into several dosage forms such as nanogels, nanofiber matrices, nano emulsions, etc. And here we can directly use these nano dosage forms in personal protection equipment like gloves, masks, coveralls, and in personal care products like sanitary diapers, sanitary napkins, like those things. And here you can see the methodology of the synthesizing of this metal-doped silica nanoparticles. First, we add 10 milliliters of tetraethyl orthosilicate and 5 milliliters of glycerol into 1.5 metal salt solution. And then nitric acid is added dropwise to adjust the pH to 1.5. Then solution is tied for 3 hours by adding dropwise ammonia to adjust the final pH volume to 8. And then the gel was centrifuged, allowed to age in ethanol for 72 hours, and then it is oven dried for 1 hour and calcined for 5 hours in 500 Celsius temperature. And finally, we characterized the synthesized nanohybrids, and also we evaluated the synergistic antimicrobial activity. Here you can see the mechanism of the synthesis of these metal-doped silica nanoparticles. Tetraethyl orthosilicate and metal salt solution are hydrolyzed in this acidic solution, and these intermediates are formed, and then they are condensed in alkaline medium and formulated metal-doped silica nanoparticles. And after synthesizing the metal doped silica nanoparticles, we characterize these nanohybrids to confirm the successful synthesis of metal doped silica nanoparticles. First, we perform the XRD analysis to confirm the crystalline structures in metal doped silica nanoparticles. Here you can see there are 3 spectra. Those are governed for silver doped silica nanoparticles, copper doped silica nanoparticles, and cobalt doped silica nanoparticles. Here you can see their respective peaks are well matched with their respective JCPDS files. So this confirms the successful synthesis of metal doped silica nanoparticles. So then we conducted FTIR and Raman analysis to further analyze the bond vibrations, peak positions, and their shifts due to this doping of metal nanoparticles into the silica nanoparticles. So first we perform FTIR analysis in the full range. As you can see here, the hydroxyl bond vibrations and the silica oxygen bond in silanol network has shifted to right by resulting a red shift in all the other metal doped silica nanoparticles. And then we analyze the fingerprint region crucially to investigate the metal oxygen bond vibrations. Here also you can see the relevant metal oxygen bonds in the spectra of silver doped silica nanoparticles, copper doped silica nanoparticles, and cobalt doped silica nanoparticles. And then we perform the Raman analysis to further analyze the bond vibrations. Here you can see the Raman spectra of silica nanoparticles, silver doped silica nanoparticles, copper doped silica nanoparticles, and cobalt doped silica nanoparticles. The silicon oxygen vibration at 640 in silica nanoparticles has shifted in other spectra, and also we can see here there are so many responsible metal oxygen peaks in other spectra also. So we can confirm the successful synthesis of metal doped silica nanoparticles by these analysis procedures also. And after that, the XPS analysis was performed. And here you can see the XPS spectra. XPS survey analysis also show the relevant binding energies of various electron configurations. Here you can see the in the XPS survey analysis, graph A is cobalt doped silica nanoparticles, graph B is silver doped silica nanoparticles, graph C is copper doped silica nanoparticles, and graph D is silica nanoparticles. And then this XPS, after this XPS survey analysis, it was further analyzed by the XPS narrow analysis. In graph B here, you can see the oxygen 1s and silica 2p electron configurations here. Here also you can see the binding energy has shifted to left side because of the doping of these metal nanoparticles into the pores of the silica nanoparticles. And then you can see down here the deconvoluted high resolution XPS spectra of silver 3D state, copper 2p state, and cobalt 2p state. These results also confirm the incorporation of silver, copper, and
Can I
cobalt.
just disturb you?
Yes,
Why
sure,
don't you
madam.
use a cursor? You point it out. Use the
Okay,
arrow.
sure, madam.
Ah,
Yeah, sure.
yeah.
So here you can see the deconvoluted high resolution spectra here. This is the deconvoluted high resolution spectra of silver 3D state. This is the deconvoluted high resolution spectra of copper 2p state. And this is the deconvoluted high resolution spectra of cobalt 2p state. So these graphs also confirm the successful incorporation of silver, copper, and cobalt metal nanoparticles into the silica nanoparticles. And after that, here you can see the bond vibrations have shifted to left because of the successful doping of metal nanoparticles. And after that, we perform the morphological characterization of a metal doped silica nanoparticles. In the here we perform scanning electron microscopic imaging and transmission electron microscopic imaging. In the images of silica nanoparticles, here you can see all the particles are spherical in shape. And in this image, you can see there are some light color spaces there. So these are the pores of the silica nanoparticles. And in this high resolution TEM images, you can see them clearly, these light color pores. So the task here is we dope the silver, copper, and cobalt nanoparticles into these light color pores. In these images, the metal doped silica nanoparticles, I think you can see these light color pores because our metals have doped into these pores. Here in the dark spots, you can see the doped metal nanoparticles into the silica nanoparticles. So these images also confirm the successful synthesis of metal doped silica nanoparticles. And in these HRTM images, here you can see the D-spacing. D-spacing means the means the distance of the lattice fringes of the each metal nanoparticle. So these values of d-spacing is well matched with their d-spacing values of the XRD spectra also. So it also confirms the successful incorporation of metal nanoparticles into the silica nanoparticles. And then we perform the EDS analysis. In these graphs, you can see the relevant peaks and the compositions of each metals and each atoms in each sample. And in the mappings, these EDS mappings, here you can see the homogeneous distribution of each atom in each sample. In these EDS mapping samples. So these images also provide clear evidence of the successful incorporation of these metal nanoparticles into the pores of the silica nanoparticles. So after that, we perform double S analysis to confirm the dope percentages of each metal into the silanol network. Here you can see the dope percentages, and they do not show a significant difference between their main percentages values. So they are we can say they are statistically equal, and it is important because we formulated bi and trimetallic silica nanohybrids by considering these doping percentages are equal, and we mix them in equal portions by mechanochemical grinding, and we formulated bi and trimetallic silica nanohybrids. And after that, we perform DPPH radical scavenging assay to evaluate the radical scavenging activity of these synthesized nanohybrids. Here you can, as all of us know, generating reactive oxygen species is the main mechanism of metal nanoparticles, which governs for the antibacterial activity and also antimicrobial activity. So when considering these results, here you can see the highest radical scavenging activity have shown by the trimetallic silica nanohybrid of silver, copper, and cobalt silica nanohybrid. And the second highest radical scavenging activity have shown by the bimetallic silica nanohybrid of silver and copper nanohybrid. And when we are talking about the monometallic silica nanohybrids, the highest radical scavenging activity have shown by the silver doped silica nanoparticles. But when we comparing all the results, we can clearly see here the best radical scavenging activity have shown by this trimetallic silica nanohybrid of silver, copper, and cobalt doped silica nanohybrid. And then we perform
Can I disturb you? How many more slides?
There are 5 minutes.
5. You have to hurry up. Huh? Be brief.
Okay. Then we evaluate the antibacterial activity of synthesized nanohybrids by using 4 bacteria, 2 gram positive bacteria, Staphylococcus aureus and Streptococcus pneumoniae, and 2 gram negative bacteria, E. coli and Pseudomonas aeruginosa. Here, first we determine the minimum inhibitory concentration and minimum bactericidal concentration values. Here you can see the trimetallic nanohybrid of silver, copper, and cobalt of silica nanoparticles have shown the minimum value for minimum inhibitory concentration and minimum bactericidal concentration, which confirms it's the highest antibacterial potential. And then we perform and then we calculated the LC50 values of our synthesized nanohybrids. Here also you can see the trimetallic nanohybrid of silver, copper, and cobalt of silica nanoparticles have shown the minimum IC50 value, which means it shows the highest antibacterial potential. And then we perform the well diffusion assay for all the tested microorganisms.
Will you sum up your findings? There is no time. We have just 15 more minutes. One more presentation. Please sum up your findings.
Okay, here you can see the number 4 disk. It is the trimetallic silica nanohybrid. It shows the highest zone of inhibition. Here I have included one graph for your easy understand. And here in green color, the highest inhibition zone you can see here, which was shown by the trimetallic silica nanohybrid. And we extend our study to clinical isolates also. We have chosen 5 clinical isolates for each microorganisms. And here also
you're going
you can
on.
see.
Sorry to disturb you. So you could have
It's
just
okay.
shown the parameters in one chart, all the different parameters, instead of showing one photograph like this and taking so long. That's not the way you present in session like this. You have to get ready for 10 to 12 minutes maximum. Okay, conclusion. That's good. And your slides are all the time too big. Half of the things are outside the screen. Okay, tell us your conclusion.
Okay,
Please
so
tell us your
we
conclusion.
concluded that. So we concluded that trimetallic silica nanohybrid of silver, copper, and cobalt of silica nanoparticles shows their significant synergistic activity, both tested gram-positive and gram-negative bacteria, and also it covers an extensive coverage of gram-positive and gram-negative bacteria as a new hope for emergent broad-spectrum antibacterial agents. So thank you very much.
Okay. Thank
So I
you
would like to
for your
acknowledge my
presentation. I think next time you are still very young. Next time you should be more careful, time yourself properly, make your slides properly so that it doesn't go out of the screen. Yeah, that's another important thing which we have to look into. I think there are few chats. I think we can just take up one or two. I think what is the degradation rate of nanosilica? Somebody has a Dr. I think it's Dalal's site. Degradation rate of nanosilica. Will you
So still
answer?
we are not studying about the degradation rate. So still we are not studying about the degradation state. Actually, this is a part of my research. So we are conducting the degradation test also. So after that, we can say how the degradation rate of the silicon nanoparticles.
Okay, fine. Any more questions from the panelists? If none, I think we have to thank you. So you want to ask question? Okay, please go ahead.
Maybe I missed. What was the control that you used? When you
Yes, for the so you mean for the well diffusion assay?
No, yeah, for the last one, when you were seeing the antimicrobial activity on the different bacteria.
Yes, I have used erythromycin for gram-positive bacteria and gentamicin for gram-negative bacteria as the positive control, and the negative control is ultra-pure water, which I have used to make these metallic nanoparticle dispersions.
So my understanding is that when you combine all the dopant with all the metals, I think that combination is the best. Is it?
Sorry, madam?
You may, no argument, please. Please write it down, your opinions, Dr. Rituparna. Okay, I think with this, we have to thank you. And we go to the next presenter. There's no time. We just have 12 minutes left now. The next presenter, who is going to be the last presenter for this session, is Dr. Sylvain Dongmo Muapi. I hope I have pronounced your name rightly. We all are from different parts of the world. I think it's so. She is a PhD scholar from Cameroon, and she has been working on herbal extracts, including in epilepsy. I think with this brief introduction, may I ask you to make your presentations? Please go ahead, make your presentations. Dr. Moapi, Dongmon Moapi. Okay. She was there, she's disappeared.
Maybe let's wait 2 minutes to see if she comes back.
She's not presenting?
Maybe there was some problem with connection on her side.
Okay, she was there actually.
Yeah, yes,
She's disappeared. So I think some network problem?
maybe.
Hmm. Do we wait? What do we do? She's the last speaker of the session.
I would say let's wait a bit.
Okay, we'll wait for a few minutes, right? 1 or 2 minutes, because everybody should get a chance. She was there, actually.
Maybe when we wait, maybe 5 more minutes, you can ask, you can answer any question which maybe wasn't answered in the list.
I think the previous speaker also has disappeared possibly. So she's there. She's there. What do we do? You
We
tell
can-
us what we are supposed to do. Whatever you tell us, we are going to do.
I would say let's wait 4 more minutes to see, but maybe
She
you
was
can.
there actually. As soon as
Yeah.
I started announcing her name, she went off the screen.
Yes, I saw it.
Mm.
Maybe you want to answer a
Maybe.
question or
Vicky, Dr. Inani, any more questions or any more answers? Your work is very, very, very good actually with the upcoming antibiotic resistant organisms. And this could be because of rampant use of antibiotics to the animals.
Mhm.
Is it because of the rampant use of antibiotics for the animals? Okay, I
Did you
think
ask
there's a
me
lot
about?
of network issue, I think. Yeah. Only I can see me and Ritupurn. Nobody else. Okay. You tell us. It's interesting. You may hit you must work some more so that you may come up with something very important issue for the world.
Okay, I'm here. We
So
are
is it
waiting
because of the
for
rampant
Marelli.
use of antibiotics for the animals to treat their or to prevent infection?
Do you mean the use of the antibiotics for animals?
Yeah. Is it rampant use
Actually, it's
or
required.
random? Whatever way you want to use it, say it.
In our country, it's random, especially for the antibiotics for
Random.
both human and
Preventive.
animal. It's random.
Is it preventive measures?
Yeah. Nowadays they are starting to make some preventable measures, but actually from very long time it's on counter. Anyone can buy antibiotics and anyone can use for even human or animal, or even they can put in the water for the farm.
Mm-hmm.
Dr. Marelli already joined us. I think now she can hear us.
So she joined back.
Yes.
I think the network issue, right?
Yes, network is disturbing. I wish to know if I can still present.
if it's okay to present at least 5-7 minutes, because we that would be fine.
Can you help me sharing my screen?
You can try with share screen if you press on it, click on it.
Yeah, please open your PowerPoint first on your screen, and after that you have to click the share screen, which we can see as green color here. That's what I did also with mine.
Come again, please, Prof.
You have to open your PowerPoint first.
Yes, it's
in your
already
desktop.
opened.
Yeah,
Yes.
then click the share screen, which is green there, down there on this screen. On this screen, on Zoom link here. This one.
Oh, okay.
You click this.
Worried. Okay, save.
I think your cursor is somewhere up there. Okay, you clicked it. Click and see.
Yes.
We have the
I
expert
think
technical help also is there. She is very good. Yeah, parcel is coming up.
is that
I think your
okay?
network is slow. I think internet is slow.
Yes, internet.
Mm-hmm.
Yes, we can
Yeah,
see.
it's coming up.
But
Is that okay?
yes, please
Yeah,
note.
yeah, it's coming up. Only internet is slow.
Please note it's only 5 minutes, and then we'll have to close the session to open a new session.
Yes, you tell us the materials, methods. Very brief
Okay.
conclusion. I think we finished with that.
Okay.
Material methods
I
and your
think
conclusion.
yes, material and method. Briefly, I can say that we collect avocado,
You do the slideshow. You are not doing
garlic,
the slideshow.
and
Make the
turmeric.
slideshow. Do the slideshow. From your computer,
I'm
click
not
the
getting,
slideshow.
Prof.
From your computer, you have to click the slideshow.
Yes.
If you can put your slides on the full screen, maybe.
Yeah, it's coming now. Oh,
Okay.
If not, we can also go through slides like that. That would also be fine.
This is the methodology study. Is that okay?
you tell us your materials, not the method. Just tell us the material and the conclusion.
Okay, all right. Briefly, we prepared methanol extract from
materials
avocado,
and the conclusion.
garlic. Yes. Okay, right. Okay. Rapidly prepared. We used to prepare avocado, garlic, and turmeric from collected in our area, and then usually they are used as fruit or spices. We prepare the different methanolic extract and we tested against the Saccharicida stages. We also evaluated the antioxidant activity and the cytotoxicity activity of our plant extract. Rapidly, as conclusion, we can say that the different path plants we tested has a good activity against the Saccharicida, against the Saccharia stage of schistosoma parasites. Also, the methanolic leaves, stem bark, and roots of the different extract possess a good antioxidant activity. Particularly, they are able to scavenge the free radical produced during the disease. Also, was it that our different extract possess a low cytotoxicity effect. It means that they are safe to be consumed in our environment as we usually do to fight against the disease. We can also say that the different plant extracts are rich in secondary metabolic like protein, phenolic, cardiac glycoside, and alkaloid that can justify the activity of our plant extract. So in one word, as we don't have time, that's what I can rapidly say about the work we conducted here in Buea in Cameroon. Thank you.
That's all. Okay, you've been very kind.
Yes.
Complying with our request.
I've
I have
tried.
to thank you for that. And it would have been very interesting if you could have present a whole of your paper. These are the common,
Yes.
garden kind of vegetables, which we eat day in and day out. So it would have been very interesting for every one of us. And I say thank you very much for that. We can take just one query, hot question, if anybody wants to ask, just one. Will you allow us one question, if anybody? Technical stuff, or is it the time is up?
Time is up, unfortunately.
Okay, I think we have to say thank you very
Okay.
much to all the
Thank you.
speakers and all the
Thank
panelists,
you.
and also thank you for your technical help. Thank you very much.
Thank
Thank
you.
you. Thank you very much.
Thank you so
Have
much.
a nice day.
Thank you.